Now let us move towards another topic in the bacterial genetics, that is transduction. So transduction is the transfer of a portion of DNA from one bacterium to another bacterium by the bacteriophage. So bacteriophage is a virus, and in the transduction there are mainly two types of transduction. One is generalized transduction and another is specialized transduction. So generalized transduction occurs due to the packaging error, whereas the specialized transduction is an excision event. So before going towards the generalized transduction and specialized transduction, I want you to know the life cycle of the bacteriophage virus. Basically, there are two types of the life cycle in the bacteriophage virus. First one is virulent or lytic life cycle. So in the virulent or the lytic life cycle, the large number of progeny phases are built up inside the host bacterium, which ruptures to release them. So this is basically the lytic cycle, because there is the rupture of the bacterium to uh, release the viruses that are packed inside their capsules. So in the non-lytic life cycle, what happens is that, so non-lytic life cycle, the host bacterium is not harmed and the phase DNA becomes integrated with the bacterial chromosome as prophase, so which multiply the synchronously with the host DNA and is transferred to the daughter cell. So this process, the non-lytic cycle process, is also called the lysogeny, and the bacteria harboring prophages are called lysogenic bacteria. So in the lysogenic bacteria, the prophase we have like an additional segment of the bacterial chromosome coding for the new characteristic, and the non-lytic cycle, or this process by which the prophase DNA confers the genetic information to the bacterium, is called lysogenic or phase conversion. So after knowing the virulent or non-virulent and non-lytic life cycle, or lytic and non-lytic life cycle of the bacteriophage, now let us proceed towards the individual, generalized and the specialized transduction. First of all, let us discuss about the generalized transduction. As I have already said that the generalized transduction is the packaging error. So the lytic phase infects the bacterium, which leading to the cleavage of the bacterial DNA. The parts of the bacterial chromosomal DNA may become packaged into the phage capsids, and the phage infects another bacterium, transferring these genetic materials. So these genes are transferred after infecting the new bacterium. I am going to describe the step by step of the generalized transduction. So for in the first figure, you can see there is the bacteria which have its own DNA. So the bacterial DNA I have denoted by the blue color, and the bacteriophage DNA I have denoted by the red color. So the bacteriophage infects the bacteria and it transfers its genetic material after the infecting to the bacteria. So uh, after the lytic phase, that means bacteriophage infects the bacteria, there is the cleavage of the bacterial DNA. After the cleavage of the bacterial DNA, you can see there are both the viral DNA, I mean the bacteriophage DNA, and the bacterial DNA are freely floating inside the cytoplasm of the bacteria. So what happens that after the cleavage of the bacterial DNA, the bacterial DNA gets packed into the capsules of the bacteriophage. The bacterial DNA gets packed inside into the capsule of the bacteriophage, whereas the bacteriophage DNA also gets packed into the capsule of the bacteriophage. So there are many capsules. There are, I have shown only 3 here, but there are thousands of capsule which consist both packages of the bacterial DNA as well as bacteriophage DNA. So what happened? The cell membrane of the bacteria gets ruptured and there is release of the new phase from the lys cells. So in the lys cell there is release of the both bacteria. Uh, there is the release of the lys cell. The lys cells may be the capsule containing the genetic material of the bacteria also, or the lys cells could be the genetic material of the bacteria also. So these could be the any capsules. So after the release of the new phase from the lys cells, it infects the another bacteria. So after infecting the another bacteria, the genetic material that has been acquired from the previous bacteria is released inside the new bacteria, and after releasing the genetic materials into the new bacteria, this new genetic material gets incorporated into the bacterial DNA. So the gene transfers to the new bacteria is seen as the end product of the generalized transduction. So in the generalized transduction, there is the transfer of the genetic material of the one bacterium to the another bacterium. There is no any changes in the previous bacterium, but there is change in the newly transferred bacteria. So this is the generalized transduction. Now let us move towards the specialized transduction. Now let us discuss about the specialized transduction. The specialized transduction, as I have already said, that it is an excision event. I'm going to describe brief in the beginning, then I will go in this step by step. So the lysogenic phase infect the bacterium, and the viral DNA incorporated into the bacterial chromosome. So when the phase DNA is excised, flanking the bacterial genes may be excised with it. So the DNA is packed into the phage capsid and can infect another bacterium. So the newly produced bacteria will be the different from the donor and the recipient. That means the genes that is present in the new bacteria will be different from both the donor and the recipient. So now I'm going to describe in the detail about the specialized transduction. So first of all, let us describe in the first step. So what happens is that the bacteria is infected by the lysogenic phase. So the phase bacteria have denoted its genetic material by the red, and the bacterium genetic material I have denoted by the black. So this is the first bacteria. So it is the first bacteria is infected by the lysogenic phase. So the viral DNA will be incorporated into this bacteria. After the infection, the viral DNA gets incorporated into the bacterial DNA. So what happens is that the DNA, the newly formed DNA will be the containing both the genetic material from the bacteria as well as the lysogenic phase bacteria. That means bacteriophage. So we have the genetic material from the lysogenic phase bacteria that is incorporated into the bacterial DNA. So after the cleavage of this bacteria, the phage particle carry the bacterial DNA, and it is packed into the lysogenic capsules. So there are the various bacteriophage capsule inside the bacteria, and those capsules contain the both previously infected bacterium gene as well as the bacteriophage gene. So after what happens, after the rupture of this bacteria, there is release of new phase from the lys cells. This new phase contains both the genetic material from the previously infected bacteria as well as the genetic material from the virus. So this phase infects the other bacteria which have new genetic material. That means the genetic material from the bacteria in this bacteria is different. I have denoted the new bacterium genetic material by the blue color and the old by the black. And you can see is that the bacteriophage which infects this new bacteria has the genetic material from the both the old bacteria, that is denoted by the black, and its own viral gene that is denoted by the red. You can see here. So after the infection, the genetic material of this previously infected bacteria and the new bacterial virus gets incorporated into this new bacteria. So this new bacteria will have the new gene. The new gene will be totally different from the donor and recipient. You can see in this genetic configuration, it has the red portion of the DNA, the green portion, the blue portion of the DNA, and the black portion of the DNA. That means the new bacteria will have the genetic material of this original infected bacteria and the genetic material from this first lysogenic phase virus that had infected the first bacteria and it has its own itself. So it will be, the gene will be totally different from the donor and the recipient bacteria. So this is an excision event that happens in the specialized transduction. So now let us move towards the transposition.